on, which is part of the homework. Please let me know when you receive it.
I haven't received for now.
You have not?
Yes.
I hope you receive it and it's not too big of a file. Is there anyone who has received it or not yet?
Okay, I think I received it already.
Okay, good. I'll share, in any case, I'll share my screen and you'll be able to see it from my screen as well. So, today we have to start with some theory. So, is this okay that we start?
Yes, you can start.
Remember at the very first class that we had together, we talked about the different isotypes. And one among these isotypes, there was the mu-M isotype. Today, we start off by seeing how, at the molecular level, we can synthesize this heavy chain mu, or M, immunoglobulin. And if you pay attention to this PowerPoint slide, everything will be clear to you. So basically now, you should all be fully aware that at the chromosomal level, at the germline level, we have several V genes. You see them here with green. At the last class, we had a look at the structure of these variable genes at the germline level. And remember, we talked about this L-part1. We said that there is an intron that you see here. There is a second exon, and there are many more features that we had a look at through the databases as well. So basically here there are two different variable genes represented. There are some D-diversity genes, they're the joining genes, and then they're constant genes, the constant exons. This is the DNA that has not been rearranged yet. This is at the germline. And I'm not sure if you remember, but we had briefly mentioned, we had said that the rearrangement that takes place at first is between the diversity and the joining gene. So you see it here, rearrangement, DJ. First is this rearrangement, and then there is a second rearrangement. There is one V with the product of DJ. Therefore, we'll have this outcome. This is the rearranged DNA, where we have one V, one D, and one J gene, as well as the constant genes. Now we have the pre-messenger RNA. You have all molecular biology knowledge, so I suppose it's quite straightforward. There is a polytail tail that is added, and this is then, there is the so-called splicing. You all know what a splicing is. It's the excision of introns and the different exons that they get together, and we have the messenger RNA. We have the translation, that is one codon, 3 codons 3 nucleotides, 1 codon, 1 amino acid. This is at the protein level. This is the polypeptide chain. We have the elimination of the signal peptide, and then here you see we have the heavy chain mu. Actually, have a look at how many constant regions we have got. There are 4. If you remember, when you were presenting, or at least the group that they were presenting the structure of the immunoglobulin heavy mu chain, there were 4 constant regions. And we had said, depending on the number of the constant regions, we have different isotypes. Is this slide clear, or do you have any questions on this? Hello?
It is clear.
It is clear. Thank you. Actually, it's quite straightforward, I would say. If it's you follow the steps of molecular biology, the central dogma in biology, we go from DNA to RNA to protein. It's only, as we said, it's multigenic families. There are several V, several D, several J, and there are 2 rearrangement processes that will take place. The first one, DJ, I repeat. The second one, V with DJ. And then we have the pre-messenger RNA, we have the RNA, we have the elimination of the leader peptide, this L here, and then we have the heavy chain. This is the basis. Now, I'll introduce another mechanism, which is called switch. You see it here, the name, in uppercase letters, in capital letters, switch. Switch is like, you know what the switch is in English? Switch is when you turn on and turn off the light. This is a switch. So this is in analogy with a switch. In molecular biology, you can switch from one isotype to another isotype. So basically, this mechanism will allow us to go from one isotype to another isotype. It will allow us to go from instance from IgM heavy chain to IgG heavy chain. And I'll explain it to you. Have a look at this, where I have my cursor, the very first line. This is, from the previous slide, this is a rearranged DNA. So basically you see that the rearrangements have taken place here, and we have the constant region. We have all the constant regions. And have a look at this arrow here. This is straightforward. This part here describes precisely what we saw at the previous slide. The generation, excuse me, the generation of a mu heavy chain. So it's exactly the same as the previous slide. I repeat, we have the messenger RNA, and then we have the translation, we have the peptide, we have the heavy mu chain. Here, maybe, I don't know, now you have the slides as well. You see some little signals. I could make it a bit bigger. Oops. It's not what I want to do. Hold on a moment. I wanted to make it bigger and I changed the slides. I'm here. I wanted this one. And I want to make it a bit bigger so you get to see the different. I want that you see, well done. I want you to see this one, SMU, which is a signal, which is a feature within the sequence. And I would like you to see this other kind of little bluish or light blue color dots, S or little circles, or S gamma 3, S gamma 1. These are signals that they are found within the locus at the sequence level, and these are the ones where splicing can take place. So if we follow this time this arrow, I'll make it smaller again. I'll make it normal, that is this time. We follow this arrow down here. You see that there is SMU S gamma recombination that has taken place, and I'm not happy with the way I see at my screen. Do you see it clearly yourself or not? Hold on, Mama. Stop sharing and I'll start all over again. Share screen. Do you see it clearer now?
Yes, we can see it now.
Right. So basically, you remember that we just saw the different signals, SMU, S gamma 1, S gamma 3. And then there is the so-called switch, or alternatively, it is called recombination SMU, S gamma. In that case, S gamma 1. If there is a recombination between this S gamma 1, which is this one, that is all this part here will be eliminated, will be excised, will be taken away. And we will left with this IGHG1, that is this part here. And you see here, SMU, S gamma 1. Then the same procedure will happen. There will be the messenger RNA, and then there will be, with the poly A tail, there will be the excision of the leader peptide, and then we'll have the G1 heavy chain. Is this clear, this slide? This slide shows us how to go, how precisely, how on the molecular level, we can go from one heavy chain to a different heavy chain. How we can go from the M or mu heavy chain to a G heavy chain. And I repeat, there's this signal, SMU, S gamma 3, S gamma 1. Of course, you realize if there is a recombination between SMU, S gamma 3, then we'll have the IgG3 isotype. Because remember, for the immunoglobulin heavy chains, the isotype of G, there are more than one subclasses, okay? So there is, in this slide, there is the SMU S gamma 1 recombination, and there is the IgHG1 chain that is being generated. Is this clear?
Yes.
Right. You might be asking me, so why do we need to know all these details? All these details, they're extremely important for people who work in the field of immunogenetics. And anything that has to do with drug development, or more precisely monoclonal antibody, therapeutic monoclonal antibody, people, they need to know these details, because they will have to choose between, for instance, if they have better efficiency with one isotype versus another one. And now you know, because we saw last time, there within the databases, the IMGT GeneDB database, you can have all this information. This is the annotation that we talked about. And today, last time you did a bit of the annotation because we went through the database and you could, one of your classmates showed us how, where exactly the coordinates, the start and the end of every feature was given in the database. You'll do a similar exercise today for these signals, for these motifs, in order to better understand the theory. So basically, you might be able to find where is this SMU or the S-gamma-1 signal. What else do I want to show you? Right, I want to show you this that I think you know already. Oh, sorry, I put it to the other. The poly-A signal, I'm not going to spend extremely, how can I say, I'm not going to spend much time on it. You all know what a poly-A signal is. I don't think that there should be more time spent on this. However, I would like to spend a bit more time on this one. Remember yesterday, no, the day before yesterday, sorry, we were talking about this VRS. Does anyone remember what is this VRS?
The recombinant site.
Well done. It's the recombination signal. It's the variable recombination signal. This is the JRS, the joining recombination signal, and this is the 5' diversity recombination signal, and this is the 3' diversity recombination signal. I have a question for you. Why do you think there is only one recombination signal for the variable and the joining genes, whereas there are two recombination signals for the diversity genes? It's easy. I'm sure when I give you the answer, you will say, yes, of course, but you know it. Do you understand my question? Should I repeat my question? Guys, is my question clear, or do you want me to repeat it?
Can you repeat the question, please?
Sure. I'm saying that we have recombination signals, and my question is the following. Why do variable genes have got only one recombination signals? Joining genes, why do they have only one recombination signal, while diversity genes have got one and two recombination signals? Why do they have two recombination signals, the diversity genes?
Because the diversity region is between the variant and the joining region.
Yes, you're really close.
So, like when first the diversity and the joining arrangement, so it needs to have a recombinant signal there, and then next after that, when we have the DG gene, then we have to recombinant again with the V. So, like we need another recombinant signal between the V and the D2.
Perfect. Well done. Exactly what you said. And look at this. We'll take it a bit further. We had talked about the nonamer, the spacer, and the heptamer. Remember, we had said the nonamer is 9 nucleotides, the heptamer is 7 nucleotides, and then we have a spacer, which is either 12 or 23 nucleotides. And there is this famous rule of 12-23. That is, you see here that we have put it cannot recombine. The V cannot recombine with the J because the rule of 12-23 is not respected. There is a first recombination of the D with the J where the rule of 12-23 is respected. And once this has been created, there is a recombination of the V with the DJ complex where the rule of 12-23 is respected once more. Is this clear? Do you understand what are
Sorry,
these?
can you repeat again?
Yes, sure. I can repeat the rule, or I can repeat from the very beginning.
Just the rule.
The rule.
Yeah.
Remember, before yesterday, we were having a look at the recombination signals. And we said a recombination signal consists of 3 units. What are the 3 units? There is the heptamer, there is a spacer, and there is a nonamer. The heptamer consists of 7 nucleotides. Then there is a spacer, which is either 23 or 12 nucleotides. And there is a nonamer, which is made up of 9 nucleotides. And then I tell you that there is this famous, in quotation marks, rule that a recombination in the immunoglobulin genes happens only when the 12 and 23 rule is respected. And I'll try to explain this rule. If there are 23 nucleotides in one spacer, and if there are 23 nucleotides in the other spacer, a recombination cannot take place. So you see, a variable gene cannot recombine with a joining gene. On the other hand, there is 23 and 12, the spacer distances here. A J gene can recombine with a diversity gene. And that is what happens at first. They recombine at first. Once this has happened, then a variable gene can recombine with this complex because the rule of 23 and 12 is respected. Is this clearer?
Okay, thank you.
Right. I have, I would like that we discuss about the different splicing sites. This, please pay some attention because we'll have a question on this as well at your homework. You have a donor splice and an acceptor splice. I think you might already have heard about these terms. A donor splice is always with GT. What's the G? Which sort of nucleotide is the G? Come on, guys, you know. It's a guanine. The G is a guanine. The T is a thymine. Adenine, guanine at the acceptor splice. So we always have GT at the donor splice. We always have AG at the acceptor splice site. So far, so good? Is this clear, this one?
Yeah.
Yes.
Right. What I would like to describe at this table here, the different splicing types. There is splicing type 1, SF1, splicing frame 1, SF1, splicing frame 0, SF0, splicing frame 2, SF2. So the question we will have will be equivalent to which splicing frame is it? And you should be able to understand and give the correct answer. How will you decide which among the 3 splicing frames is the correct one? The answer is here. We said that the donor splice should be GT. The acceptor splice should be AG. You realize what's happening, right? There is some nucleotides here that they will be deleted. The GT and the AG will be deleted. And look what happens. The previous nucleotide, the nucleotide of the 5' to the GT and the nucleotides on the 3' to the AG, they will get together to form one codon. Of course, we know that we need 3 nucleotides for one codon, because one codon will give rise to one amino acid. This is the genetic code, and I'm sure that you all know about this. So, and if this is the case, if we take one nucleotide before the GT and 2 nucleotides after the AG, we have the splicing frame 3. If there is no nucleotide, I mean, if we are at the correct reading frame and we continue, and there is nothing, there is only the GT and the AG, and then this area is being deleted and the splicing is it continues normally, then we are at the splicing frame 0. If there, I don't know if you see it here, if there are 2 nucleotides at the 5' of the GT and 1 nucleotide at the 3' of the AG, then we have the so-called splicing frame 2. Is this clear? All this information for the genes that have been already annotated within IMGT, they are found within IMGT Ligma DB. This is the work of the biocurators. Within IMGT, we employ biocurators. There are people like you, more or less, that they have finished their Master 2, and then they do extremely detailed work on the sequences of the different immunoglobulins and T-cell receptors. There are people who have got knowledge of immunology, and they have some knowledge of computer work as well. Currently, IMGT has got approximately 20 people working currently, and the biocuration team, I would say it's a bit less than half of the people in the team. The rest, they're bioinformaticians, and they're like informaticians as well. We have master students, so we have, of course, PhD students and postdocs, as any other academic team, that they work on research projects. But there is also the database platform, which is much more engineering job, and the biocuration team. So basically, the different types of splicing, I hope they're clear. You always need to find first the donor splice, then the acceptor splice, and you need to look, this is what's going at the 5' and what's going at the 3' of this GT and AG respectively. Here, as you see, this is the intron sequence that will be excised, and this is the 5' of the exon, and this is the 3' of the exon. Depending on how many nucleotides, if any, we use from the 5' and how many, if any, we use from the 3', then we have the different splicing frames, be it 1, 0, or 2. Okay? Can I go to this a bit more theory? Do you have any questions, or should I cover this for the first part, this last slide?
You can continue.
Perfect. So, I'll go ahead to describe how at the molecular level we can have a heavy mu or m either secreted or membrane heavy chain. How do I start? I start off with this one. This is the line that we'll have a look at. At least the very first part should be extremely familiar to you right now, because we are at the rearranged DNA. You see what has happened. We have 1V, 1D, 1J. They have rearranged. And then we have the constant regions. You see CH1, constant heavy 1, constant heavy 2, constant heavy 3, constant heavy 4. And look at this. There is CH, S for secreted. There are two different polyadenylation sites, and there is M1 and M2. This is how it looks like at the genome, the rearranged genomic sequence. And have a look at this arrow. We're going to see how a membrane mu or M chain is being generated. So basically, there is the transcription. We go all the way to M2, and there is this polyadenylation signal that is maintained. You see it clearly here. This is the transcribed, this is the transcript with the polyadenylation signal. Then when there is the translation happening, of course, the leader peptide will be excised as usually. Similarly, the poly A tail, and this is the peptide. This peptide contains the 4 constant regions as well as M1, M2 that allow this membrane heavy chain. You see it here. Now, if we take this arrow vertically, this time we'll go all the way up to here, to the internal splicing site. And as you see here, we'll have the 4 constant regions. We'll have the CHS, the secreted part, and we'll have the first polyadenylation signal, this one. There will be the translation. The leader peptide will be excised. This signal here will be kept. The constant heavy secreted. And at the end of the day, we'll have the generation of a pentamer, 5 mu chains on a secreted form. Is it clear? Basically, we play with the 2 poly-A sites, the 2 polyadenylation sites, and these are M1, M2 that they will give rise to membrane heavy chains, and the CHS for secreted will give rise to the secreted pentameric form of the mu heavy chain. Is this clear? During your homework, you'll be asked, for instance, to identify the CHS. Of course, it will help you through the database, to go through the database and identify where precisely it is located. The idea of the homework that you'll be asked to do now, it is also to familiarize you with the theory. The idea is to go back to the slides and see, what is this CHS? Is the secreted element that we discussed about. What is M1, what is M2, and where are they found? It will be much clearer when I show you the homework. So I stop sharing this one, and I ask you please to open this file, this Word file. I ask you. Actually, it's the very first that I showed to you when I started in Montpellier. So it's from 2013. I just changed this. It doesn't matter because this is the basics. They have not changed the basics and they are all always the same. I just took away the date. So this is the file that you should all have received. Your name, your first name, you're not M1, you don't need this. Right. I would leave it as simple as that. Right? Guys, are you with me?
Yes.
It's only that I sent you the very first file. I did not update. You can update it with me as I update it here. I tell you, these are the basics, and the basics have not changed since 2014. They're exactly the same. And what I will ask you, I would like all of us to do this first exercise, domain C. This is a constant domain. This is, for instance, the first question. Give the name of the IMGT gene and allele. To facilitate your life, I have given, after the questions, I have given the sequence. So basically, you can retrieve it from the databases, but you can work here. I would highly recommend that you retrieve it from the database, even if it takes a bit of time. This is why I have put it at the bottom of these questions. And here you see this is the accession number of the sequence from LinkDB. And actually, this is the accession number that you find the sequence everywhere. It is the date of the sequence. It's a sequence that was sequenced the 3rd of May 1906. I told you, I took a sequence that is kind of confirmed. It was updated in 2013. I had taken a recently updated sequence. It's a human immunoglobulin C constant, and there is the mu and delta heavy chain genes. It's a genomic sequence. It gives you some extra information. It's from which species? It's from Homo sapiens. It gives you some different references from PubMed, where this sequence was retrieved and described. And then it starts giving you the C cluster. I don't want to give you the answer. The first question is, which is the gene or the allele in this sequence? What would be your answer? Are you with me, guys?
Yeah, we hear. Can you repeat your question?
The question is, you have a lot of questions here. You have 1, 1.1, 1.2, 1.3, 1.4. The idea is, this is your homework right now. I would like you to do this exercise. And in order to be sure that you will have no issues to do this exercise, I take the first one, 1.1, and we discuss it together. So the first exercise, okay, it's in French, but it's not difficult. It says to give the IMGT gene and allele name of the sequence you're looking at. So I'm asking you, what is the IMGT gene and allele? Do you have any idea? Can anyone share his? I'll stop sharing. Share your screen, please, and show us. If you look at the, you look at the Word file, and you should be able to find the answer. Guys, I'm waiting for someone to share his screen, please, with the same Word document that I was sharing with you. I need a volunteer.
Dear professor, I can share my screen. But
So in that case, I give you share screen options. You can share. Please go ahead. Right? So what is the gene and what is the allele that we'll be working on in this exercise? The gene is Ig, immunoglobulin heavy M. And what is the allele? Exactly, the allele is the third allele. That was the answer to the first question. Could you please show us what is the second question? Go to the top of your file, right? The second question. You need to identify a color, maybe blue, for instance, the exons. How many exons do we have in a constant heavy M chain? We have 4. So we should not do it only for the coloring. We are not only at kindergarten. We do it only to understand the theory, to make it a bit clearer, the theory. Instead of my asking you an exam and asking you how many different CH domains do we find in an M heavy chain, which will be much more difficult, I provide you with this exercise. And if you do it properly, I suppose it will be a bit easier to register this information. So basically, you should go further down to your file. Yes, find the CH1. Oops. Yes, excellent. Where's the CH1? Be a bit more specific. What is the start and what is the end position of CH1? Yes? Speak up, please. The start position is at 145, and then position of the constant heavy 1 domain is at 456. Great. How do I find it in the sequence? Where do I find the sequence? If you go...
Okay. I'm
Yes?
sorry, my mic was off.
Okay.
Okay.
So
you want to ask the CH2 position?
no, we have not finished with the CH1. I'm asking for the CH1. What are the positions of the CH1? Please repeat.
The position is start at 145 to 456.
Perfect. So where do you find these positions? Where do you find 145 to 456?
I think I will find it in here, the sequence here.
Yes, please show us.
Here's 120, then
There are 10 nucleotides.
130
30.
and then 140,
40.
41, 42.
Excellent. So basically you can 45, 145, and then you'll go up until
here, this one.
Correct. And then up until where?
Excuse me?
And what is the end of the CH1?
The end position, 456, 400,
Exactly.
30, 40,
There are 10.
51, 52, 3, 4, 5, 6, this one.
Right. Excellent.
56.
So basically you can choose this area in a Word document and color it blue. Correct? You can do it afterwards at your own time, but you have understood how you can, where you can identify and where you can find your sequence. Correct?
Yes.
Right. If you go further up to see the other questions now. Right. So basically the question was to find the exons. And now the CH4 CHS, you might remember, I hope. If you don't remember, you'll go back to your PowerPoint slides and you will see that the S is the secreted. And then you'll find the CHS, right?
Yes, I think I can find it.
You'll do this. I think it's easy. You go to the third question. Let's see what's the third question. If you go a bit further up. The questions. The third one, it asks you to find the stop codon on the 3' of the CHS. Actually, here it's it should be B, but it's C, doesn't matter. It tells you to identify the internal splice sites within the CHS, to identify the polyadenylation site as well. Then it asks you to find the exons M1 and M2. I'm at 1.4. It asks you to find the stop codon on the 3' of the M2. It asks you to find the splicing site of the donor and the acceptor between M1 and M2. This is the question that sometimes the students have got difficulty with, the D. All the others, I think they're simple questions. It asks you what is the type of the splicing, and this is the PowerPoint slide that we had a look with the 3 different splicing frame types, and it asks you why. It asks you to find the second codon stop at the 3' of the M2, and then it asks you to find the second polyadenylation site, if there exists, and if things exist. So I think that you should all be able to tackle this exercise. Do you have any immediate questions? Do you understand what I'm asking you to do? You'll become biocurators. You'll become biocurators with work that has already been done. So you have your questions, you have the sequence at the bottom. I highly recommend that you take the accession number and you retrieve the sequence from LinkedDB as well. If we do it simultaneously, all of us, a bit less than 20 people, it might take too long. If you start doing it slowly, each one of you, eventually you'll get your sequence through the database. In any case, you have most of the information at the bottom of the questions, and then you can start answering the questions. Is this okay? And please, you'll write your name, your last name, your first name, you'll do the work. You're not well, perfect, as you have done it. You'll do, maybe, Anne, you don't need to leave this deuxième séance 14th of October 2014. You can get rid of this by any, yes, there. All of this line, you can get rid of this, as you can, I should have done it myself. This is, I'm sorry, doesn't matter, this as well. You do not send it to me today. You'll do this first exercise. How much time do you need for this? What's the time now? Or at least you can start working on it, and when you are done, I don't know, you need 30 minutes, 40 minutes, you need an hour. Guys?
I think if we need further stuff to do, we can keep it in like a homework and we will submit you tomorrow, and you can continue another task for this. Is this okay? Or you still waiting for me for us to complete this exercise?
It's up to you. Either I can go to the second part, because if you go further down of this Word file, there is more things to do, or we can do it step by step. Actually, for tomorrow, I had a suggestion as well. I sent you an email. Did you get to see it?
Yes.
Right?
It's like conference or something like that.
Hold on a moment. I'll share it with you. There is this conference happening tomorrow, a bit earlier than today. It starts at 2.5 hours earlier than our class. But there is the Asian Bioinformatics Consortium Symposium, and it's online. So I had the suggestion that we could all attend this symposium, because it's Asian symposium. You're in Vietnam, there will be talks from China, there will be talks from Japan, and there will be talks from Korea. And they are in the bioinformatics. They're about different databases, which I think it would be of interest, and you'll get an idea. And I was I was thinking of that we all go to this, and the class tomorrow will take, if you don't have any objection, we'll take another kind of time slot, the week of 19th of December onwards, and we have another class together. What do you think? Are you interested in attending this?
I think it's okay for me.
To me, this is extremely valuable for you because no matter what you do, you'll have a bit more broader idea of bioinformatics, and then you'll see a bit of what's going in Asia. You might ask me, are there other conferences? Yes, of course, there are other conferences as well. There are a lot of conferences in bioinformatics. However, I thought that that was extremely of interest, and it happened, as you see, I got it the 7th of December. It was announced recently. It's a small consortium. It's happening online, and it's an Asiatic bioinformatics conference, which is much closer to home, to your own home. It's not a European bioinformatics consortium. So if we are all fine with this, we say that tomorrow we attend this conference. And then we can see for the week of 19th of December to have, to replace or to see how we are getting on, have another class together. Is this okay? I only heard one okay. Are you available the week of 19th of December?
19th of December.
The 20th of December, for instance. Are you available?
20. The drop class have learn to learn all the morning and the afternoon, and the biomedicine we don't have the class on this day, so I think it's not available first.
So which date is available for you? Are you available on the 19th of December or the 21st of December? Are you available on the 22nd or the 23rd? Which days are you available?
I
think it's
21st is okay.
The
Afternoon
21st is okay.
Yes.
What's afternoon? Afternoon for you, afternoon for me?
Afternoon for me. It's the same time with our class today.
Okay, so the 21st, is this okay if we say that we have another class on the 21st of December?
Yes, I think it's okay for us.
For all of you?
Yes.
Perfect. And we say, hold on a moment, we say that tomorrow we attend this conference. And what I would like from each one of you, I would like that you take some notes. And I would like that you have like one paragraph or half a page at the end where you put bullet points, what you thought they were the interesting points from the different conferences that you heard and that you attended. So basically, what I'm trying to say, I do not want you just to get into this conference and I let the people talking and I don't know, I play around or I cook and I don't listen, it doesn't matter. I would like you to be actively listening. Now I understand, you might tell me, but it's much longer and this and that. Okay, I understand this. So you can choose one conference. If you cannot start from the morning, from the very top till from the very start till the very end, choose one section and listen to it carefully or as carefully as you can and try to take some notes. And the idea is that we will try to discuss these notes afterwards to see what we learned from these conferences. Is this okay?
Yes,
Yes,
it's
I
okay.
think it's wonderful.
Perfect. So this is for tomorrow. I try to be there. For me, I'm sure I will be there. I'm not sure I'll be from 6 a.m. onwards because it's 6 a.m. my time, which means I have to get up at 5 a.m. I have the kids at home as well. I don't want to wake up the whole place at 5 a.m. We'll see, but I'll be around. In any case, we sign up and we follow the Zoom conference. It's online, right? So now, I don't know how we should do it. If you want, I can get to the second part of this file without you having done the exercises. If you see at your Word document, I can share it myself. I was doing it slowly. I was doing it step by step. What do you see? I'm not sure what you're seeing now. Hold on a moment. I'll share the Word file. From the pedagogical point of view, it would have been nicer that you finished this exercise before I ask you to go to the second part, because the second part in your Word document is this exercise 4 that we're going to get to see. And these are the rearranged sequences. So you should have understood, or you should have covered the genomic sequence before we get into the rearrangement. And I don't want you to mix these things. So I'm thinking aloud now. No, we're not going to do this. What I will do, you start a bit your exercises, and even if you don't finish, I'll prepare to give you the homework that I had in mind for next week. So I'll give you the you'll start doing your homework, the questions that I just said, and I'll prepare the homework that I had for next week. So in 10 minutes or so, I'll interrupt you and I'll show you the therapeutic monoclonal antibodies. And I'll ask you the homework. However, I don't want any panic. This homework, it's not going to be neither for today, nor for tomorrow, nor for the very first Monday, the 12th. It will be from Wednesday, the 14th of December, or even Thursday, the 15th of December. We'll discuss it together. Is this okay?
Yes, it's okay.
Perfect. So you give me 5-10 minutes, you start working on the questions, and if you have questions, you'll get back to me, and I'll interrupt you in 10 minutes maximum to show you what we'll be working on. I do not want to start exercise 4. I don't think from the pedagogical point of view it's good to do it right now. I would have liked that you finish the exercise 1, all of you first, we discuss it, so you have a clear idea of the genomic, of the chromosome level, the germline level before we go into the rearrangements. I don't want you to mix them. So
Okay,
start
so
looking. Correct. Start looking at your exercises in case you have any questions. You'll ask me, and I'll bring out and I'll send you even by email the other information. Hello, everybody. I sent you 2 email messages. Can I explain to you? Can we have a little? Can you stop what you are doing, and then I'll explain a bit to you what you're supposed to do.
Yeah, okay.
Yes, okay.
Is this okay? So basically, I'm going into the, maybe I share my screen. It will be easier as well for you. Share screen. You see the main page of IMGT. You know about this, imgt.org. And within the databases, I'm clicking on this database, the last one in the list, which is called IMGT MapDB, monoclonal antibody database. Right? I click on it. Should make it a bit bigger, maybe. So you see, anyway, and this is the interface of the database. There is an abstract that describes the database. I can choose randomly. Tell me which antibody you want to choose from the database. There are a bit more than 1,000 different antibodies. I don't know. I take randomly this one and I search into the database the information about this specific monoclonal antibody. And this is the information I retrieve. First of all, I have an ID number. This is the one I gave actually, the 481, because each item in the database has got its unique number. There is INN, International Non-Proprietary Name. This is an international, as the name says, instance that deals with the naming of the antibodies. And this is based in Geneva, in Switzerland. And there is an expert group in this consortium that gives, for instance, dapirolizumab pegol, the name of this antibody. There are some lists that they are being put together, and IMGT biocurators will go and extract these lists once they are published by the INN, and will do the annotation of them. So basically here, you get the INN number for this specific monoclonal antibody. You get the list number, all this, the recommendation list, the common name of this antibody. It gives you the species. It's a humanized antibody. That is, the only thing that is not from human is the CDR, the complementarity determining region. And then if we have time, this will be the very last project that we'll work on. This is the humanization of an antibody. So basically, imagine that we have a mouse that has been affected or by an antigen, has been affected by an antigen, correct, and it has developed antibodies. And these antibodies, we realize that they are extremely efficient against this antigen. We would like to keep this efficiency for the equivalent human disease. However, we do not take the very same mouse antibody because there is high risk that it will be rejected by our body. We only take these small regions, the loops that we discussed, the three CDRs, CDR 1, 2, and 3. CDRs, they stand for complementarity determining region. You can forget about the name. But as we said, they're the regions that they bind to the antigen, especially the CDR3. So we'll keep the mouse CDR and the rest will be human sequence. Therefore, we call it a humanized antibody. Here, you see the format of this antibody, and I would highly recommend that you take this format from the database for your homework. I'll get to what your homework will be precise, but you can take advantage of this format of the antibody here. It's a standardized format for all the antibodies, monoclonal antibodies in the database. It gives you some extra information. This is a Fab, G1, kappa, that is a light chain. It is conjugated with Pegol. It gives you the information into this database. There is a cross-link. We have not explored this database yet. It gives you the specificity. There you are. It's not a mouse. It's a rat original sequence, origin clone sequence. It's Ratus norvegicus. It's a rat sequence. And maybe I put this to the other side. It gives you the company name that has created this therapeutic monoclonal antibody. It gives you the clinical indication. It's for systemic lupus SLE, basically. I have difficulty in pronouncing erythematosus. So basically it's the lupus. Usually we say the lupus disease. It's an autoimmune disease. It's at the clinical phase 2, and the clinical domain is immunology. So basically, through this database, you can have access to all currently known therapeutic monoclonal antibodies. And what is, I sent you 2 emails. The first email I sent you has got an Excel file. I'll open this Excel file and I'll try to share it with you. It's this MABS Excel file. Enable editing. I'll share it, and basically you get to see it, right? It has got a list of different monoclonal antibodies. There are more than 500 antibodies, and the idea is that each one of you chooses at least 2 monoclonal antibodies from this list. Right?
Yes.
Okay. I continue for the... Then you have a results file, a PDF results file. I'll share this document as well. You see, for instance, this PDF file, we are looking at this. Can I make it bigger? Is atezolizumab. This is the monoclonal therapeutic antibody. I repeat, this, the format, it's easier to take it directly from IMGT MapDB the way I just showed it to you. And what is the request for you? The request is to study the mechanism of action of at least 2 different therapeutic monoclonal antibodies. You are supposed to go to different web pages, the different web pages of the pharmaceutical companies or the biotechnology companies that they develop these monoclonal antibodies. You can have a look at articles within PubMed. I suppose you all know PubMed, right? You can extract publications. Several of them, I would say they're open access, they are free. You can access them freely. There are some journals that you have to pay. Do not pay, please. Ask me in case you really want an article, and I'll see if it's accessible via the University of Montpellier library to give it to you. So basically the idea is you find your sources. I will not help you find the sources. This is your task. You're at the master level. I'm sure you can manage this extremely well. The idea is it might take you some time. You find an article that clearly describes the mechanism of action of the antibody that you're interested in. You can choose any sort of disease indication. It could be cancer, it could be autoimmune diseases, it could be anything you want to. And then you have to create a graphical representation of the mechanism of action. Something equivalent to this one. For instance, you see here what, and actually, hold on a moment, I have, and this is the only thing I ask you at your report. You'll send me, each one of you, you'll send me a report. The report will have a graphical representation and a little bit of a text, like a paragraph, like a legend, like a legend, like a figure description of this graphical representation. I shared you another email message, the second email message, where I have an article. Please keep it confidential because we have not published it yet. It's fresh out of the oven. We just finished it. We have not submitted it yet. It will be submitted next week or so. But I send it to you because we worked on these monoclonal antibodies, and I would like to show you. I'll share it with you once more. Share screen. I'm sharing, for instance, one figure of this article where you see clearly, I'm sharing the figure 2. You see ipilimumab, the different actors, the different players, the different types of cells. And you see, for instance, there are T cells, this ipilimumab that binds to CTLA4. And once there is this binding, the co-inhibitory signal is not in place anymore. Therefore, there is a stimulation signal, and this stimulation signal has got a higher proportion of T cells that they are being generated. There is proliferation of T cells. These T cells, they will release cytokines and they will target the cancerous cells and eliminate cancerous cells. So basically, I'm giving a really rough description right now. I would like you to come up with something equivalent to this, a graphical representation with the figure explanation below. I give you an Excel file, I repeat, of a bit more than 500 different monoclonal antibodies. I would like that you work on at least two completely different mechanisms of action, each one of you. I would like that each one of you works on a completely different monoclonal antibody. I don't want all of you to have worked on the same monoclonal antibody. Please coordinate the efforts among yourselves so you work on different monoclonal antibodies. Additionally, I would like you can have a look at the article. This is why I share it with you. You can have a look at all the figures. However, I would like you not to present any of the monoclonal antibodies in the article that I shared with you. Obviously enough, because I would like you to do the work and not take it straight away from this review article. I have shared with you the Affinity Designer software little documentation. This is the software that we used within IMGT to generate these figures. This is, however, not compulsory. You can use either this software, Affinity Designer, which is free for 30 days. You're not going to spend 30 days to do your figures. In any case, you're going to present your work next week. So you can use Affinity Designer and I have given you some steps to understand how to use it. I had the feedback from some students that they found it a bit difficult and not so intuitive for users. I give you the permission to use any other software that you are familiar with or that you would like to use. Some students, they opted to use BioRender because they had used it beforehand and they found it more easy to use. You can use anyone you want to, and I would like from you a report. A report, it could be either in a PowerPoint slide, or it could be in a PDF, or it could be in a Word document, or it could be any way you want it to be, any format you want to be. It's open the format. However, please, a format that I can view. And the last comment, sometimes there are different antibodies that they have the very same mechanism of action. I do not consider them as two different mechanisms of action. I consider them as one mechanism of action. I repeat, I would like you to study at least two different mechanisms of action. You could have studied 5 or even 10 different monoclonal antibodies, but if 5 of these antibodies, they have one mechanism of action and the other 5, they have a different mechanism of action, I consider them as two different mechanisms of actions. Is this clear what I'm asking you to do? Is this clear, your project?
Yes, it's clear for me.
Okay. And you think that...
I want to ask that.
Sure.
That each student has to at least two monoclonal antibody to work with. So do we have to compare our two monoclonal together, or we just list the graph and give the legend for the graph? Do we need to comparison to monoclonal antibody we have? Do you need we have to comparison to mechanism of two monoclonal antibody we choose?
Right. You do not need to compare the mechanism of action. You need to list the mechanism of action of your monoclonal antibody.
Yes.
They might not be easy to compare. They might be completely different targets. I do not ask you to compare them. I ask you to present in a way that it's understandable by everyone how your monoclonal antibody, the one you have chosen, works at the healthy or at the cancerous level. In case we are looking into a monoclonal antibody in oncology domain. If you have a look at the review that I sent to you, I think it will be crystal clear the way that I would like you to work on your mechanism of action, which is similar to the one described in this review. Please note, I do not want you to write an article. No, I want only you to present graphically your mechanism of action. So if you have extremely difficulty in doing it digitally with a software, take color pens and color it. I don't mind. And then scan your picture or whatever, like at kindergarten with different colors, blue, red, as you do with your exercise. I don't mind that much. However, please try, because we are in bioinformatics. So it would be nice that you manage to. It would be nice that you manage to figure out how a software tool for graphical representation works. The important part, the most important part, is not the graphical tool. The most important part is to understand the mechanism of action and to go into MapDB and take the antibody and present it in a way that the others can understand how this monoclonal antibody works. And the only thing, you need to coordinate among yourselves, I repeat, so we do not hear the very same antibody 20 times. We hear 20 different antibodies. Okay?
Yes, it's okay.
My question is, when do you think, or when would you be ready to present this work? I would say that you have to send me your report, your PDF file, with at least 2 mechanisms of action, and each one of you presents 1 mechanism of action orally. And you ask one little question to the audience. Once you have presented your monoclonal antibody, you can ask a little question to your audience about what you just presented. So, when would you like this to be? Do you think you'll be able to do it on Wednesday the 14th or Thursday the 15th?
I think yes, we can ready from the last lecture. It's 16th Friday. Is this okay for you?
The Friday 16th, we might replace this day as well, because there will be some internship students. Do you want to do it on the 21st of December?
Yes, I think the 21st of December is better.
Okay, so 21st of December, I'll have it for presentations. Is everyone okay with this?
So I want to ask that how long we have to present the
How
time
long?
for each person. Yes.
You present, how long will it take to present? 3 minutes? 4 minutes? It's up to you to give us the summary of the mechanism of action of the antibody you have chosen. It's not long. It's good luck. Sometimes this thing that translates, it does not translate properly. It is not gonna take you that long to present.
So we're going to present on 21st December. So when will we have to send you the PPT?
You can present on the 21st of December and send after the end of the class and after the end of the presentations, you send me your PDF or your PowerPoint. Don't forget that I want to have the resources, that is where you found this information, as well as a little text describing what we see. Like the figure legend, as I told you. If you have, I repeat, if you have a quick look at the article I shared it with you, you'll understand exactly what I mean. It will be much clearer. The resources, it could be, what is the scientific article that you read in order to give the mechanism of action? Do you know it by heart? I don't think so. I don't know all the 1,000 different monoclonal antibodies by heart. Far from it. Is this okay?
Yes,
Fine.
I got it.
Right? Most of the times, I give students some time to work. You can work in pairs, or you can work 2 of them together. If you work 2 of them together, that is, you'll have 4 mechanisms of action. I don't mind that you collaborate among yourselves, because each one of you will present a completely different mechanism of action. Fine?
Yes, I
Okay.
understand.
Perfect. So basically, you have another hour. You can finish the homework. Tomorrow, we get at this Asian conference. On Monday, the very same time, we'll start off by correcting your homework, and then we'll tackle the exercise 4, which is the rearranged sequences. Okay?
Okay.
We'll see them on Monday. You'll have a bit of work to do on Monday, and then we'll do the humanization that I mentioned Wednesday and Thursday. Friday, I'm not sure we'll have a class, I repeat, on the 16th of December. We'll see what happens. And then on the 21st, we'll have your presentations. Any questions?
Excuse me, that do we have to finish the exercise 1 to 3 and send it to you?
No, you have to finish the exercise, all the exercise 1, 1, 1, 1, 2, 1, 3, 1, 4. You have to finish it, and we'll sit together on the 12th of December.
Okay, thank you.
However, I'll ask you to present it. Therefore, please don't say, I don't have to send it, therefore I will not do it. You have to have done it. Is this clear?
Clear.
Perfect. Do you have any questions so far on your exercises? I don't want anyone to tell me, I did not understand anything, I did not do it. If you don't understand something, you can even email me. I would like that on Monday, you have all finished the best you can do for the exercise 1. If it's clear, then I let you work on this, I let you continue this, and I see you tomorrow.
Okay, see you tomorrow.
Take care. I'll announce to the organizers that there will be some students as well, so they don't get what's going on. And I'll see you tomorrow.
See you tomorrow.
Take care. Bye-bye.
Okay, bye bye.